folks. It's Dr. Gilchrist here. I had to stop recording yesterday largely because of the room that I was in recording here got a little bit too hot and my mic basically wanted to fail on me. So I am finishing up our section on validity and experimental control today. You will also notice I have chopped off my hair. Yeah, we're not going to be getting haircuts for a while, and I really badly needed one, so I did it myself. It is not abjectly terrible, so we'll kind of roll with it. So, the last time that we were here, I was laughing my head off about Your Baby Can Read. And one of the things that I mentioned is that when you're watching these videos, and I would highly encourage you, if you find the time, go onto YouTube, look at some of these videos, and look at the kind of signals that parents are sending to their kids to help them pick the correct answer. And when we see this happening, it is not because of babies learning how to read, certainly not from the same stimuli that they are trained with over and over again. Rather, what you are seeing are what are known as demand characteristics, which kind of go hand in hand with expectancy effects or observer bias. If you already know what kind of data you're looking for, if you already know what your hypothesis is, then you expect your participants to behave in a certain way. And you might send them signals for how they should behave. And here's the thing. Participants do have implicit norms for how to behave. Generally, they want to be a good participant or a good subject. So they are unconsciously going to behave in ways that kind of help you out. And so because of this, it's really, really important not only to do blinding, as we mentioned before, making sure that anybody who's running the experiment is unaware of the hypothesis you're trying to test, but it's also why experimenters do not reveal a hypothesis or the purpose of the study until it's over. But even when you do this, you may unconsciously be providing cues that indicate how participants should respond or what the study is about, and those are demand characteristics. So you're going to behave in these subtle ways that basically bring about the results you're looking for. And it's not necessarily because of the experiment itself. It is because you have been sending signals to your participants, hey, this is how I want you to respond. And then they do. So, how do we avoid demand characteristics? We can do, your book talks a bit about manipulation checks. Another way of talking about this is what is known as a suspicion probe. We can do a post-test questionnaire to see if participants knew what the true purpose of the study was. So, for example, last year in our replication study, we were trying to see if priming people with stereotypes of older adults, such as words like old, gray, Florida, helpless, and so on, would cause people to walk more slowly. Now, of course, we didn't tell people about the true purpose of the study. We instead told them that what we were doing was basically looking at language processing abilities. And so one of the ways that we tried to make sure that we knew that participants didn't guess the true purpose is we gave them all a post-test questionnaire, basically asking them, hey, what did you think the study was really about? And most of them realized they guessed that the purpose of the study was to look at language processing. So from my perspective, I feel like we did a pretty good job of not really sending those demand characteristics out. Now, here are some other things that you can do if you want to avoid these demand characteristics. You can increase the psychological realism in the task. You can make it more engaging. If you make it really psychologically engaging for people, they're not really going to be thinking about what the true hypothesis of the experiment is because they're too involved in the experiment. We can look at pilot testing. So we can test run this on a few people and see if they figure out what the purpose of the experiment is. We can use unobtrusive measures, such as things like accuracy or response time. We can look at hard to distort responses. You might potentially want to avoid a within subjects design in these cases. particularly if you are diverting what the true purpose of the study is, the more that you do a within subjects design, the more likely it is that the participants will be able to guess what the experiment is about based on the different conditions that they're exposed to. So within subjects designs can work, but if you're utilizing deception or if you're worried about demand characteristics, don't do it within subject design. The other thing that you can do is what we call the good old red herring. You can deliberately mislead your participants with what the purpose of the experiment is, just like we did, telling people that we were looking at language processing when we were actually looking at a priming study. So another thing that we have to talk about, if you are utilizing drugs of any kind, we need to take into account a placebo effect. So anytime that you are looking at an effect of a drug, whether it is a new pharmaceutical treatment of some kind, or if we're talking about an alcohol study, there are going to be some biological effects of the drug itself, but there are also psychological effects. So, for example, when I was in graduate school, we actually had an alcohol research study group on campus. And by and large, their studies were by far some of the most popular studies to participate in. Why? Because depending on the group that you were in, you got to drink beer. Now, I will mention that to make sure that that this lab didn't get in trouble with the law, you did have to be 21 and up to participate. And you were subject to a breathalyzer to make sure that you were not intoxicated when you left. And one of the ways that they would try to help metabolize the alcohol a bit faster is they'd give people pizza. So these studies were quite popular because you got pizza and you got beer. But here's the thing about alcohol. We all have a pretty good understanding of what alcohol does to a body, and we have certain expectations about what it's going to do to us. So anytime that we are taking a drug, we are not only experiencing the biological effects of the drug, we are also experiencing the effect of our belief in the drug. So I could potentially give somebody a non-alcoholic beer, and I don't necessarily tell them that it's non-alcoholic. But they might actually believe that they're drinking alcohol, and as such might report psychological effects as if they were drunk, despite the fact that there's no way they can actually be drunk. There's no biological effect here. So if they're reporting feeling drunk, it's because of their beliefs or because of psychological aspects. So if we want to make sure that a drug treatment actually works, we need to be able to tease apart the biological effects from the psychological effects. So what we will often do is include a placebo control group. So in these alcohol research studies, you couldn't give one group beer and give another group water. The beer group, they might show differences in cognition or whatever you're studying them on, not because of the actual effect of the alcohol, but because of their belief that this is what alcohol should do. And you can't tease those effects apart. And so water would not be a suitable comparison group. What you need to do instead is give a placebo. So in this case, in the beer study, it would be a non-alcoholic beer that does not actually produce a biological effect. So any effects that you see from this group would have to be psychological. So if we give one group beer and another group non-alcoholic beer, and we find that there's no difference between the two, we know that there's no effective alcohol on this. But if we do see that our alcohol group looks different from our placebo group, we'll know that it's specifically because of the biological effects of the drug itself. Now, oftentimes when we're doing a placebo study, when we're utilizing a placebo, you need to use what is called a double-blind procedure. This means that the participants shouldn't know what they're getting ahead of time. And additionally, the experimenter should not know what they're getting ahead of time. So if you know that one group's got the alcohol and the other group's got the non-alcoholic beer, you are going to run the risk of treating them differently. So it's important that the experimenter doesn't know, and the person that is taking part in the study doesn't know. So double-blind procedures basically get rid of expectancy effects, and it also helps us rule out whether or not we're seeing a placebo effect. And generally, this is something that will have to be done as a between subjects design. Now, I want you to imagine that I am doing a research study. So I'm doing a memory study. This is another effect we're going to talk about. So let's say that everybody scored 100% on the memory tests. Is this actually a good thing? In my case, if I have everybody getting 100% correct and nobody's made any mistakes on their task, that's actually a really bad thing for me. And this is what is known as a ceiling effect. You can't make any sort of conclusion when everybody gets the maximum score. There's no sensitivity. So if I gave y'all an exam and y'all all got 100% on it, I didn't do a sensitive enough test to figure out what you know and what you don't. And additionally, I've made it so easy that I can't even tell who grasps these tougher concepts and who doesn't. So a ceiling effect can be really problematic because there's no sensitivity there. And unfortunately, for us to learn things about psychology and for us to learn things about the world, we do need to make sure that we have people making mistakes. We have to make sure that people are not performing perfectly because it doesn't tell us anything. Now, likewise, it would be just as bad if the task were so hard that everybody got everything wrong. Again, it doesn't tell me what you're capable of doing and what you're not. And this is what's called a floor effect. So ceiling effects are pretty bad. Floor effects are also bad, both for the same reason. Neither of them are sensitive enough to tease out what people are capable of and what they're not. And we need to be able to do that if we want to actually learn anything about a particular psychological phenomenon. So how do we avoid a ceiling effect or a floor effect? Well, first of all, you can do pilot testing. Run a few participants, see if there's some variability. And that way you can figure out if you need to make the task a little easier. I've sometimes had to do this with my older adults when tasks are too difficult for them. Or if the task is too easy, we can make it tougher. And generally what you are going to find is that what you need is what we call a level of desirable difficulty. The task needs to be easy enough that your participants don't get frustrated and quit, but not so easy that they're not applying themselves and being challenged. And we don't want everybody to get a perfect score. Generally, I tend to find for a lot of my working memory studies, I don't want them getting 100%, and I don't want them getting 0%. This tells me nothing about their working memory abilities. Generally, baseline performance should be about 75%. That means it's easy enough that most people can do pretty well, but it's not so easy that they're not being challenged or learning anything or exerting. They actually do have to exert some effort to do this. So let's kind of move on. I thought this was a pretty interesting part in your chapter. What if you don't find anything in your experiment? And I can tell you that this has happened to me occasionally as well. One of the very first research studies that I ever did was part of my senior honors thesis looking at thought suppression. And I did a direct replication with extension. So I tried to replicate the study and I did not find anything. And this was after spending hours transcribing data, programming data, analyzing data. We got nothing. So what happens when you don't find anything? What does it mean?